You can share your screen. Yeah.
You can see that, right?
Yes, I can.
Okay, so my name is Katerina. I am from Cardiff University and so I'm a senior lecturer there. And I'm going to talk about in vitro fertilization and embryogenesis. Maybe it's a bit ambitious that I have put both together. So the idea, as I discussed with Maria, is of course to generate discussion. So I'm not only showing results that we have completed, but also things that we want to complete, and we have questions, and we're open to collaboration. So continuing, the outline of my talk is what is in vitro fertilization, and then there is a strong focus on my work over the last few years on calcium signaling and how that interacts with mechanics. And that theme is important for IVF, and it's important also for the later stages of embryogenesis. In that connection, we have developed mechanochemical models of calcium signaling that are relevant to embryogenesis. And we are in the process of doing a cell-based model. My PhD student Abhishek Chakraborty is here today working on that, and then quite a few of the other things I'm working with Thomas Woolley, as I mentioned, and Tim Oster, who I don't know if he managed to join. We had a mix-up with the hour, and he has to rush to a tutorial very soon. Okay, so starting and diving into this. So with Thomas and Tim and Carl Swan, who is a chair of developmental biology in Cardiff, have been looking at various aspects of IVF. We also set up a funded collaboration with the London Women's Clinic, and we have looked at various questions. I'm gonna look very briefly at one of them. You can see here 3 themes. One is the thermal modeling of embryos. So Maria, this is much later when the embryo has already formed and it has been frozen, because more and more embryos are frozen these days, and then they go frozen embryo transfers. And especially this was applied during the pandemic, actually, that people could not go through with their IVF cycles. So it's a very important problem to understand how the embryos respond to temperature and bringing them back to room temperature. A team has developed a new algorithm for understanding eggs, even at the 1 cell stage, so it's just an egg, and how this egg what are the flows inside the egg. And for that, you need some way to extract that flow and to understand the anisotropy in the material. I'm not going to go through that now, but if you want to know more, you can contact us and we can discuss further, especially with Tim Ostler and Thomas. And then the very holy grail of IVF is, of course, to choose the best embryo to implant to the prospective mother. And this choice, this decision, you can do, well, the ambition is that you can do it as early as possible. At the moment, they let the embryos to develop to day 5 before they put them inside the woman, inside the patient. But the ambition that Carl Swan has been really keen on is to use even at the 2 cell stage of development, can we find metrics that can decide what is the best embryo to transfer to the prospective mother? And that's challenging, very challenging problem. So just to show you the embryo that is growing back to its original size after it has been vitrified and put in the freezer. This is images from the London Women's Clinic, our collaborators. And what we have done is we solved the heat equation and COMSOL, that's easy model mathematically, but we managed to say some things to the clinicians. So we modeled the cooling of the embryos, and we asked if the freezing rate varies with the number of the embryos. And we found out that there is a minimal effect when you have more than 2 embryos. So basically the result we could tell the clinicians is that vitrifying multiple embryos does not affect vitrification rate. So they can put essentially as many as they like on that cryotop device, as they call it. And this, of course, expedites the clinical procedure, right? And it's a very pressurized environment, actually, the IVF clinic. So saving time in that way is very good. We also published a few months ago a paper where we have shown that also arranging this number of these many embryos on the cryotop device again doesn't matter. So that's good as well. And that was presented also in Fertility 2021 is the main conference the IVF experts are going, the embryologists. So coming back to this question, can we find a metric for predicting the success of the embryo even at the single cell stage or two cell stage? So Carl and team have been working for some time on calcium waves in fertilization. And this video I'm going to show you here is very interesting because it shows that there's a correlation between the calcium signaling and the mechanics. To understand the calcium signaling, you have to stain and you have to destroy the cell, but you can non-invasively monitor the mechanics. So the idea here is that can we find non-invasive methods for choosing the best embryo at the very early stage of development? So I'm gonna show you this video now. So the upper row, as you can see, is these calcium spikes. So you see this line going from left to right, and every time you pass a calcium spike, you also have a swishing motion. You have a mechanical response to that calcium spike inside that egg. And of course, as you go further, I'm not gonna finish it because time is pressing, but you can see that this correlation kind of is less and less strong as we go from left to right. And of course, the question is, can we identify this mechanochemical coupling and do a model for that and use it to predict IVF success? And what makes these differences as time progresses and that mechanics then don't really talk too strongly to the calcium anymore? And we have started working on this since 2018 that I joined Cardiff, and Thomas was there and Videria, he had met Carl, and then we had many students working on this. We must say, as we have been discussing last week, that we haven't progressed to the stage that we wanted to, but the problem is still very challenging and very interesting. So I'll tell you what we have done up to now. So before I do that, in case you don't know about calcium, it's everywhere in the body. This is what you should get from this slide, calcium everywhere. And there's a way that you can model a calcium, a standard way. You have to know a few things about the cytosol and the endoblastic reticulum. The endoblastic reticulum holds the calcium in, and there are these channels. They are the IPR channels. So IP stands for IP3, one of the chemicals that bind on these channels, and the other chemical that binds on these channels to open them and release calcium from the ER is, of course, calcium itself, which gives rise to the calcium-induced calcium release, CICR abbreviated, usually from the endoblastic reticulum. So you have a contribution of fluxes. You also have diffusion of the calcium, and I don't put here the IP3. It's also a dynamic variable, but in many cases, we consider that as a bifurcation parameter. So let's go and see how we adapted this generic idea for calcium modeling to our calcium signaling in fertilization. We have been discussing a lot with Carl, our experimentalist, Carl Swan, and he said, well, you know, I'm frustrated because there are so many models that have been done for calcium signaling fertilization, and none of them really reproduces the experimental features that we recently observed. So there were some more recent experiments. And one thing that definitely you need to do if you want to help me is to use the experimental data by Mack and al. This is, as you can see, quite old, from 1998. It's Mack and Foskett. There's another name that some people might know, very well-known experimentalists. And nobody has really incorporated this for calcium signaling in eggs. I must say here that calcium signaling changes from cell type to cell type, even though some key features remain the same. It has the generic modeling picture I showed earlier. So what we did, we thought and scratched our heads a lot, took many months, and we decided to correct some of the deficiencies, let's say, of previous calcium models. One was to remove an equation that had to do with calcium varying inside the endoplasmic reticulum, because it was shown in the experiments that this variation is very small in eggs and is not important. And we went with a gating model, which has to do with binding, these probabilities of binding of the IP3 and of the calcium to the IPR channels. So we split it up, and we ended up with a model. Well, I'll show you the model, but before that, I will show you this probability that was determined in this experimental paper by Mack and Foskett. We have, as you can see, calcium and IP3, as calcium as IP3 change, you have a bell-shaped curve here. And we incorporated this for the first time in a gating model. This is just an OND model. So in terms of analysis, it's just a nonlinear excitable model. And is, however, closer to what happens in X than previous ones. And in fact, we managed to find qualitative agreement with experimental observations. And these were that amplitude increases as IP3 increases, the IP3 concentration increases, and frequency increases too, but more slowly. And I said qualitatively because we don't have a quantitative agreement. We need to do some rescales, and we're in the process of doing that to hopefully get also the quantitative agreement. So there are many steps to go from here. As I said, this is a multi-year endeavor. We want to add the dynamic evolution of IP3 because it has observed in experiments that you also need to consider IP3 as a dynamic variable. We want to add diffusion of calcium and of the IP3, so then we'll be getting a PD model, and we probably want to add spatial structure of the ER. Here I should mention that Thomas and Carl had worked on a spatial structure model in 2018, which we also want to bring into this holistic model for calcium signaling and fertilization. So this is where we are with this. So we are excited about it, but long way from getting the full 2D, 3D model with IP3 in and everything. So we've done the first step. And then the question that I want to come back to is, how does calcium couple to mechanics? So you might know already that there are mechanically stimulated calcium waves in airway epithelial cells. I don't know if you can see, okay, yeah. Also, I'm interested in embryogenesis, and you can see here that in embryogenesis, if I play this movie from a Wallingford 2001 paper, that you can see this dramatic flash, that's an intercellular calcium wave. And at the same time, you can see motion. So these are the mechanics and the calcium that you can also see that they kind of go together. So there is some obvious coupling here. And we have been focusing with another set of people, that's Abhishek is my other PhD student, and Tim Phillips in Cardiff, and Philip Maney and Ruth Baker, and some experimentalists, Christodoulou and Skourides in the Skourides lab in Cyprus. on neural tube closure and understanding this interaction of calcium and mechanics in neural tube closure. I don't know if you have seen this picture before. It's a clearly 3D effect where you get some zipping at the end of this process and you get then the neural tube in the embryo. And if this somewhere in this stage something goes wrong, then the neural tube doesn't close, and then you can have serious malformations of the embryo, like spina bifida, which can have various degrees of severity. And it has been recently connected experimentally to calcium disruption. So going to here, our experimental collaborators, so Christopher and Spudes, published a report paper in 2015 that shows that calcium signals, I'm going to play this, precede contractions in apical constriction during neural tube closure. And if you disrupt this calcium dance, let's call it choreography of dance, then you get malformations like spina bifida and even anencephaly. Depends on the species, actually, whether it's mouse or frog, what kind of defect you get. And of course, we really want not to get these defects and to understand this process with modeling and reproduce experimental data. So, oh, sorry, I just press this again. I don't want to do that. Are there big waves here as well? Yes, there are. In later stage of neural tube closure, you do get bigger waves. So you can see a big flash here, similar to the one I've shown in convergent extension in the Wallingford work. So that's from another related paper 2 years later, quite a recent one as well, by Suzuki and al. And then we published a paper 2019 where we analyzed some of the data a bit more. And you can see clearly here how you get this dashed line is a calcium spike. And just after that, you get this contraction. And we are tracking, well, the experimentalists track the apical cell surface area of these epithelial cells. And the other thing that you can see here is that amplitude and frequency of oscillation. So the big game here is calcium oscillations, right? This is always what you want to study, because if you lose the calcium oscillations, this is when you get the malformations. So understanding their amplitude and frequency is very important. The experimental data says that amplitude and frequency increase as the cell surface area decreases. And we wanted to model this, and Murray already had thought about the problem, how to couple calcium and mechanics. You can find that in chapter 7 of volume 2, I think. Don't quote me on that because I don't remember anymore. But they used Oster and Murray bistable kinetics, whereas the more recent experimental data tell you that for calcium you need excitable dynamics. So this is what we did. So we developed this mechanical model in one dimension, one spatial dimension. And we have two bifurcation parameters. We have the IP3 level, and we have also lambda here in the calcium term, calcium equation, that it's multiplying the dilation theta. And C is again the calcium concentration in the cytosol, and H is the percentage of non-inactivated IP3 channels on the endoplastic reticulum. And the other important feature here is that we have TC, that is a contraction stress that is calcium dependent. This is directly obtained by the original ideas of Oster and Murray. I just want to check the time. Oh, sorry, I stopped. Perhaps I should go for 10 more minutes and then we have discussion for the last 20. What do you think, Maria?
Yeah, no problem.
Is that, yeah, so let me share my screen again because I lost it.
I have a question just before you go. Just for my, so calcium is, so when you look at this first cell and decide whether it's viable or not, you're looking mostly because like the cells are contracting, or are you measuring some calcium input and output? And I don't know, I didn't see the link. Is it then assumed that if it oscillates, then it's healthy? Sorry, I wasn't. So that's what
exactly.
So if you
that's
see
it.
calcium oscillating like back and forth, then it's healthy.
That's true. Yeah.
Okay.
So it's not just oscillating. It's like also the characteristics of the oscillation. But you definitely want oscillations because people have been thinking for ages, why do we need oscillations? Why this calcium oscillates? And the eminent idea is that it's because it transports information in that way. And also you want to avoid calcium to be constantly elevated at a high value because that is cytotoxic. So this is, yes, oscillation, calcium oscillation means health. If you lose this oscillation, means something goes wrong with the embryo.
Okay, that's interesting. Thank you.
So that's what we are aiming for in the model, to show that when we lose oscillations, this is what we wanted to find out. What are the conditions for losing these oscillations and hence getting the defect? So we run this, well, before I do that, I want to tell you about this bifurcation structure. So the R3 model is a nonlinear excitable 2D model, and it has a paradigm behavior that many other calcium models have, which means that when IP3 is at low levels, you get this monotonic decrease, which I haven't shown, and then you get an excitable potential as you excite more. Then you manage to get these nonlinear calcium oscillations in a regime, intermediate regime of IP3 levels. And then if you increase the IP3 more, you lose these oscillations and you get a decaying oscillation. So we need that because that's what we have tracked. We track the oscillations and we have shown in this system mechanochemical model that you get for a specific value of lambda, which is that strength of the coupling between calcium and mechanics, that if that exceeds a certain value, then you actually lose the oscillations. So this is an important thing that identifying these conditions for losing the oscillations. Let me go back to presentation mode because I don't want to go through my hidden slide. I don't want to go through the asymptotics. It's some work we did with Philip Main and John Chapman, but it's in the archive and people are interested. I'm always open to continue and finish this because it's just in a state of before submission. But anyway, we had some remaining questions there. I just gonna skip that. I just want to show also what happens when I add diffusion to the mix. Again, I remind you this bifurcation diagram, and you can see here that instead of action potential, then you get a solitary wave. That's a nice kind of tutorial thing because that's a well-established thing. And then if you have a mu 0.3, it has a calcium oscillation, but then you get a periodic wave train when you add the diffusion in. And in the place of decaying oscillation, you get a decaying wave train. And I want this because then I want to couple with mechanics and diffusion. So then I can see here that when I have no mechanics and I have a solitary wave, if I increase the lambda, I would then go to a transition, qualitative transition, and get a wave train, that's oscillations. And then if I keep increasing, this is how we visualize the loss of oscillations. You get that you lose the oscillations for lambda big enough. And the last thing I want to say in 5 minutes is that actually we extended that model with the Gator Ruiz Bayer, and we submitted it recently in 2 dimensions and in 3 dimensions, but I'm not going to talk about that. You can find it in the archive if you want to read about this. And now something that we have been working on a lot, Abhishek Chakraborty, my PhD student, has been the major person developing a code, a cell-based vertex model for this neural tube closure. And our ambition is to create a mechanochemical model. To do that, we have gone to the Suzuki et al. Work, and there they have 3 components in the model. I guess some people here are experts in vertex models. It's not actually my expertise, but anyway, Abhishek has been doing great work in understanding this and building his own code. And this code has 3 contributions. You have an energy functional, and you have perimeter elasticity term, you have an area elasticity term, and you have also the line tension. And the line tension is where you have also calcium. You just assume, and I'm not gonna go to the next slide actually to show you what we assume. Why is it not going? Yes, so the calcium effect, we model it as Suzuki did, with having a baseline line tension between these cells, which we model with hexagons. And that psi term is an addition to the baseline line tension at the interface that has to do with what is the value of calcium. At the moment, it's like a switch, but we want to obviously make that more sophisticated. So here are the 3 contributions, the area and perimeter elasticity, and then the UL is the line tension term. You have an equation for how the vertices move, and the mu there is the viscosity. And there is also a ratchet mechanism, which is what I have shown essentially in the experiments, that you get like a stepped decrease in the cell surface area as calcium comes and goes until it saturates to a certain minimum value. So I'm gonna skip all this because it's too technical and just show you some simulations from the code. So that's just 1 cell, and the calcium effect is small compared to the baseline. We can choose an activation probability. And what we are doing now, we want to see how, as time evolves and you have these calcium spikes coming up at random intervals, you get then a decrease in the cell surface area. So you do get that, but it's very rapid. So the question is, after I show you the 25 cells version, how do you vary the parameters? This is what we are at now. How do we quantify the sensitivity of this model to the 16 parameters that are involved? So another picture is when you have duration of the spike 2 seconds, activation probability 0.04, that determines the interspike interval for the calcium. You can see here the area and the perimeter. And you have 16 parameters. We have done a systematic parameter sweep, and we mapped how each parameter affects the rate of the shrinkage and the final shape and size. Actually, we find that the rate of shrinkage of the tissue is much faster than the experiment. So this is something we want to fix. So I'm gonna skip this as well. So just to give you an example, when we vary the elasticity parameter for the area, what we get is that the saturating value of the area doesn't change that much, but that Kn does affect the rate of shrinkage of the tissue. You can see the initial shape here and the final shape. You can do all the same things for Kp, which is the perimeter elasticity, and for viscosity. And it's challenging to quantify the contribution of each parameter. So we need further investigation to determine appropriate range for these values. Some of them we can be confident from the experiments, some others we simply don't know them, and we have to understand the effect it has on the model. So the challenge is to relate another challenge we have with these parameters is how to relate them to data we do know. Like we know for epithelial embryonic tissue, Young's modulus, shear and bulk viscosity, Poisson's ratio. We know this for the continuum models, we put them in. Again, there is a range there, but at least you have some values. The question is, how do you get these continuum elasticity parameters and do them discrete counterparts, Ka and Kp? Skipping that, and another challenge is at the moment the code gives instability. So I don't know if people can suggest something here if they're experts in vertex model, which parameters cause this weird shape coming up. We are not there yet with reproducing the experimental data, so it's very much an open challenge in progress. And I reached the end. I've shown something on IVF, and then I really talked about calcium and mechanics a lot. It's important for IVF and early decisions on embryo selection. It's important in trying to understand how to prohibit embryo malformations such as spina bifida. We've done some continuum modeling, and we are now doing a cell-based model. Thank you for listening, and I would like to thank all my very good collaborators for various aspects of what I talked about that we have been working together. So yeah, I hope I left enough time for people to say ideas and discuss.
Well, thank you. No, I think we still have time, and if we go over, then I will. You will become the host if, and people will trickle away at 12:30 if they need to.
Yeah. Okay.
But thank you. That was quite interesting. Is there any questions? We can start if there's questions for the presentation first, and then later we can stop the presentation and then just have our discussions. I mean, I have a few questions because this is quite new to me. I don't do a whole lot of calcium, but one of the things that I guess I was thinking about, does it matter how many cells there are, and is it like, I see it alternating? So are they like sending a signal from one cell to the next, and then all of a sudden they have like sort of a, like, is the wave between cell, or are you also calculating the wave within cell? Because when you were doing the embryo fertilization, I was imagining sometimes it's a single to two cells, but maybe I misunderstood.
So yeah. This is a very topical question. Thank you, Maria. So basically, for the big egg, you have dramatic complex system of calcium waves in that single cell, right? In the epithelial cells, you have 2 or 3 types of calcium signals. You have a single cell flashes, which we do think they are waves, but they haven't been resolved by the experimentalists. So just see this flash that comes up in the video. And that's a single cell effect again.
Okay, so flashes are not sort of, so I just assumed whenever I saw a flash, it's a spike or a wave of some sort, but maybe
it's a wave of some sort, but it hasn't been resolved. So whereas in the egg, because it's a big thing, they have measured and they have found the shape of these waves. So here we don't know the cell scale wave, so to speak. It must be there though. We do see, so this is at the cell autonomous, so you can see single cells flashing. If you go here, you see an obvious coordination between the cells because you get this flash over many of them. So that's an intercellular calcium wave. Because you have this like pools of calcium inside each cell communicates with the neighbors, and then the tissue can let this wave of calcium pass through, which is what you see here. So it's very interesting because it's a very diverse range of signals changing from system to system.
Thank you. Leah, you.
Hi, thank you for the interesting talk. I hope you can hear me.
Yeah, I can.
Okay.
Thank you, yeah.
As you mentioned, there had been a lot of calcium modeling, and it was very popular and a hot topic around the late 1980s, 1990s. And you mentioned at least one place where it was done. did you find you had to redo it, or, I mean, was the modeling in the past incorrect, or did you just have to modify it a little bit to get it to adapt it to the data? That's what I was wondering.
Yeah. So our challenge, the specific challenge, was to do calcium modeling for fertilization. So there we had to modify. It depends on how you view it. You have to modify significantly in the sense that we are based on the ATRI model, which is a very famous one. So, and then we went and introduced for the first time, let me find the mark curve, this one. So we had to introduce this one in. So the previous models for calcium signaling fertilization were lacking this curve. This had to go in. So this is what we added. So if somebody could say, yes, it's a modification of the ATRI model with this significant modification of adding that probability curve that is accurate data for the IPR dynamics, basically, that hadn't been incorporated before.
Thank you.
That's just the IVF, that's the fertilization stuff. For the other one, just to, sorry, to carry on a bit longer, because I showed the second model in case the question was for that. That one is basically a well-established model ATRI together with Oster and Murray's mechanics. So that's again modification of the Oster and Murray assumptions, but replacing the bistable kinetics with the ATRI kinetics there. Which is gonna find it. Yeah, so that's the other second model I've talked about.
Adita, you can ask questions. Everybody's free to ask. And so.
Hi, are you able to hear me?
Yes.
Okay, nice talk. I had a couple of questions. First thing is, most of the calcium modeling initially was on invertebrates and then moved to cell lines. So the data which you used belong to the embryos, or you have extrapolated that from cells?
Yeah, so we have egg data from the CARS-1 lab and other people. And we also have embryon data from Xenobus laevis frogs about this model here that is on the screen now. So it's not for invertebrates. It's
okay.
the frog
And the
here,
next one,
and the other one is the egg, the previous model.
Yeah, the second question is calcium signaling during embryo development also causes because of the mechanical changes. In your model, I have not seen that anywhere. So are you planning to integrate that as well?
Can you repeat the question, please?
Calcium signaling can also be induced by mechanical shifts, mechanical changes during the embryo development, right? So
yeah.
in your model, are you including that anywhere specific?
Yep, we are. Actually, that's a fitting slide then to answer the question, because what we are incorporate here is a two-way mechanochemical feedback. So as you very rightly said, mechanics induces calcium, and calcium induces mechanics, induces contraction. So this is a two-way feedback. So here, the way this is captured in this model is that you have one equation, the tissue dilatation equation is the mechanics equation. And there you can see that term, the contraction stress T of C. So you have the calcium fitting into the mechanics here. So I guess I should have started from the first equation though, which is the more well-known effect, where the mechanics affect calcium. And how we model that? We go to the third last term here, lambda theta over K1. And there you see that theta affects the calcium. So the mechanics affects calcium in the first equation, and the calcium affects mechanics in the first equation, mechanics affect calcium. In the third equation, the calcium affects mechanics. And this is a two-way mechanochemical feedback of this model. You have both.
Yeah, okay. Yeah. Thank you.
Thank you for the question. It's a very clarifying one.
Thank you for the talk, Katerina. I also have a question regarding the imaging and the data. Did you also work with any imaging of the cytoskeleton or specifically myosin?
For actomyosin and these kind of things, you mean?
Yes.
So we have some images. So this is this second pillar here is differential dynamic microscopy is an imaging methodology, right? So we have images of eggs, which show some flows. For example, I showed this video. Let's see here. Yeah, here. So we have this kind of pictures. And you want to extract the flow information from there. It's not at the scale of seeing structures moving. It's on this scale here that we have pictures, but you can still use imaging methodologies that deal with anisotropy, because you have obviously anisotropy here induced when calcium is in the cell doing stuff to determine and extract information from the egg.
Okay.
I think I'm not answering the question completely though. Do you want to elaborate?
Yeah, I was just wondering if you also kind of followed up on any relation between the calcium signaling and its interaction with these motor proteins.
No, well, the short answer is that we haven't. It's something we are interested in, obviously, how the actomyosin and how you model that and put it in a calcium model, and also, yeah, but we haven't done that. Are you working in this area?
Trying to or starting to, yes. So I'm interested in that, yes.
Where, in which lab are you?
I'm in Germany and Frankfurt. Yes, and we're working on a project where we're also interested in kind of these contractile waves. So that's really interesting.
Yep. Yeah, and tracking the actomyosin network and imaging that will be good. I mean, that's another level of sophistication, of course. Yeah. So we can talk more. So I mean, if you are interested in this, we're definitely interested in expanding the models in other directions. So yeah.
And also with the vertex model. So we're also working with the vertex model. So that
you're also doing vertex model.
Yeah.
You also put calcium in the vertex model.
No.
So you
that's
just do the mechanic
interesting
side of it. Yeah.
Yes. Yeah.
Good.
But now I'm thinking about it.
Okay.
You should start.
Yeah.
Thank you.
Yeah, I guess this theme of calcium and mechanics, it has been kind of overlooked. The first time we submitted that paper on saying that mechanics and calcium feedback loop is very important, then one of the reviewers really didn't like that. But really, calcium is everywhere and it really talks with the mechanics. So yeah, it's good to have that in.
So we have your email, so Zoe, you can always get in touch with Katerina through.
Yes, I will do that. Thank you.
Okay.
So I was just double checking that. If it's a different email that you you can put that there, but I have the one that we announced.
Yeah, no, it's the same one. Where is it? Just here. That's it.
Yeah, that's the same one. I'm just making sure because we don't want to end without yeah. Are there any other questions or discussions? I don't know. You had questions, open questions regarding your models. Do you want to, is there anybody
Yeah.
You want to propose that again? I forgot, sorry, what the exact question was, but there was
Yeah,
some.
this question, for example, about does anybody know about these kind of instabilities? Have they observed them in their VFX models? And do they have any ideas how to resolve them? That would be one question. If people haven't worked on this, though, they wouldn't know what to answer. It's just maybe Zoe, when we have the individual discussion, we can delve more into that.
There was somebody here who works on this, but they're not here anymore. So
Oh,
yeah.
okay. Who was that? What's the name?
I think Matt Smart is one person that I was thinking of that probably. And last week we had Rodrigo Gonzalez, so his postdoc Goncha. She also worked on she works on the vertex models and does a lot of these. So she might have had more of an idea.
Right.
I personally don't, so I don't. So I'm just, so Matt also presented. So if you look at the schedule, his email is there, and Goncha's email is also there. But maybe if they see this video, they'll hear these questions, or we can just propose it on the Slack channel. Some people might also.
Okay. Yeah.
So if you'd like to do that, that also is another place.
That would
Sorry.
be great, actually. Yeah, if people have ideas about that. Yeah.
So these,
sorry.
So these cell shapes, this what you call unnatural cell shapes, do you get that when you do your parameter regime swipe, that you go all over the parameters, or when do you observe these?
Abhishek, you're there?
Yes, I am. And that is a good question. Am I still muted? It's telling me I'm muted. Can everyone hear me?
Yep.
Right. So the thing is, what I've noticed, I did like a parameter sweep over like the set of 16 parameters. Where this pops up is if you have this sort of disconnect between like say the values of area and perimeter elastic modulus, or like if your initial sizes of area and perimeter are too different. So when the model evolves, you have a case where maybe the area is shrinking too fast compared to the perimeter. So that's what gives you these sort of sharp, acute, like interior angles. So I'm trying to wonder if I can sort of implement like a direct relation between like the area and perimeter within the model. Would that get rid of it? So I haven't gotten around to doing it yet. But this is what happens generally when there's like a disconnect between the area and perimeter, like dimensions or elasticities. Does that answer your question?
Yeah. I think there's actually some work on it. I can try to find it.
Oh, that would help a lot.
Yeah, I think what you're trying to describe, there's probably a aspect ratio between perimeter and area, and give me a second. And what you need to do is satisfy that. So if you start with hexagons, think about what that aspect ratio would be, and you probably have to keep that as they change. Exactly. And it's when you get those too far off, in order to accommodate that, you get those unnatural shapes.
Exactly, that's exactly what I was planning on doing, like the relation between the area and perimeter with the hexagons. And Clinton,
yeah, so what
I've met you, but I've written to you before. Sorry for interrupting. I wrote to you before with about the vertex model. In fact, I read your thesis and I was inspired to write the thing in Python.
All right, that's why I joined today. I had shared that before you had joined the call.
Right.
Yeah, so what I would check in this is track those aspect ratios throughout time, or throughout your simulation time, to see how those change. And if that emerges, if the unnatural shell shapes emerge when you cross some threshold in that aspect ratio.
Right. Would do that. Thank you.
And on that note too, I was interested in this, the picture you have up there right now. Do you have any fixing in space of the boundary, or is that web-like feature just purely from this shell shape change?
Right. So I don't actually. So I don't know if it's in one of the previous slides. I start with this hexagonal arrangement of cells. So what happens is I've tried before with like this more sort of rectangular arrangement of cells. So you have like these like rows of cells just stacked on top of each other. So because that sort of tissue shape has 4 corners, you get this when the tissue shrinks, you get the corners sort of sticking out. You have 4 corners sticking out, whereas in the case of the hexagon, because it has 6 corners, as the tissue shrinks, you have 6 corners sticking out. But what happens is in some cases, if the simulation like runs for like long enough, or that's the ideal case, what happens then is the tissue shrinks to this more like circular or ovoid shape. And that happens both in the case of the rectangle and hexagon if like things have gone right. In the case I have these sort of corners sticking out, I'm generally assuming something has gone wrong. It's either because the tissue is moving way too slowly, because like the forces at the corners, the corner cells, they're kind of low compared to the forces on the interior cells, which are balanced by their neighbors as well. So there's one thing I was thinking of looking into was setting different drag coefficients or at the corner cells, or maybe implementing a boundary condition. And this is like more tied to the experiments. So this is what the when I was speaking to the experimentalists about this, they said that you do get this sort of dragging effect where like the neural cells are pulling on the epidermal cells, and those are the ones that drag. And you should try to aim for a more circular arrangement of cells, so then you don't get these corners jutting out. So these are 2 things I want to modify the code and implement.
Yeah, so in my thesis, in the dorsal closure section, the way that we remedied that is we had elastic tension lines that were just fixed in space for that boundary, and those can also help to serve to help circularize those boundaries as the tissue starts to shrink.
Right, that would really help.
And so there's no mechanics on those other than they're just elastic, so they will resist pulling, but there's no active dynamics on them.
Right. That's kind of something similar to what we're doing with the epidermal cells. So like they're just there, but they are capable of moving freely. But we're not actively putting any like calcium dynamics and stuff on them. But I will look at what you've mentioned in your thesis as well. Thank you for that.
Yeah.
Any other questions or? Right. Clinton, you want to introduce yourself again, just so Abhishek can get to know you, and likewise?
Yeah, so I'm Clinton. Abhishek reached out to me, I would say estimate like a year ago, maybe a bit more. So my PhD thesis was at UBC in the math department, where I did similar vertex models on epithelial morphogenesis. I looked at dorsal closure and salivary gland formation. And so both of those are very similar to the latter part of the talk. And now I'm a postdoc at the John Innes Center in Norwich, UK, where I'm studying the cell mechanics of grass and Eudicot stomata, which are the cells that open and close to allow for respiration during photosynthesis. I think you're on mute.
Who's can you hear me?
Yeah, we can hear you now.
Yeah. So yeah, I am in the final year of my PhD. I have been working, I did my MPhil with Katerina as well at Cardiff University. And that's the time when I was looking at the continuum models that you saw earlier, and you were discussing earlier.